Classically, experimental psychology delineates three different varieties of attention. Those are depicted on the screen in front of you. First, we have selective attention, and that has to do with focusing on task-relevant stimuli. Most of the research in aging deals with the topic of selective attention, so we will clearly put the emphasis of this lecture on selective attention. However, there are two other varieties of attention that must be noted. Divided attention, that's the situation where you're juggling multiple goals at the same time and seemingly doing multiple tasks at the same time, or so-called multifunctioning, multitasking. We'll take a look at that area as well and see what happens as we grow older. And then finally, we'll briefly consider sustained attention. That is what we call vigilance, the ability to stay on task for a protracted period of time. And one of the things we'll see surprisingly is that vigilance is maintained quite well in old age. And you'll see on the bottom of the slide, there's a reference to something called the span of visual attention, or the useful field of view. And that's sort of a specialty area that's developed in gerontology and aging. And if we have time, we'll spend a bit of, if you will, of the lecture on this topic of the useful field of view. So let's begin with selective attention. And when we talk about selective attention, that is that ability to focus, to essentially separate out the task-relevant stimuli from the task-relevant stimuli, and then process the task-relevant stimuli to a higher level. We'll consider the classic experiment of Patrick Rabbit. This experiment has been replicated multiple times, and the results are considered to be a fact of modern gerontological science. So let's describe the task that Rabbit did. It's a pretty straightforward task, but it has some profound results. He did what's called a card sorting task. And on his card sorting task, he had decks of cards, and each deck had 48 stimulus cards in the deck. And your task was to take these cards and to sort them into two piles. Every card either had a letter A on it or a letter Y. And so you would sort the cards into the A pile and the Y pile, if you will. And the independent variable in this experiment was quite interesting. And that was, how many distracting elements were there on the cards in a given deck? And there could either be no distractors, there'd just be a single target, either an A or a Y, and nothing else on the card. Or there could be one distracting element, or 4 or 8. And the dependent variable in all cases, no matter which deck you were sorting, was the time it took you to sort the deck into two piles. So I have a crude drawing in front of you here that shows sort of an example of what the different decks of cards might look like. So you can see in the card represented here as deck 1, there's a single letter on the card. So it must be either an A or a Y. It must be one of the targets, because remember, all the cards have a target on them. And then in deck 2, you can see there is a target, in this case the letter Y, but there's also a distractor. And in deck 3, there's a target, here it's an A, and 4 distractors. And in deck 4, there's a target, the Y in the middle, and 8 distractors, so to speak. So there's 4 decks, and they vary in terms of the number of distractors they have, either none, 1, 4, or 8 distractors. So when Rabbit had young and older people sort these cards, what did he find? Well, again, you see this little quick hand-drawn diagram that I have in front of you, this figure. What he found was, for young people, depicted in this bottom function here, that no matter how many distractors there were, 0, 1, 4, or 8, their sort times were virtually the same. The amount of distractors had no impact upon them. Younger people could effortlessly, I shouldn't say effortlessly, but very quickly separate the wheat from the chaff. They could find the target letter and quickly make the sort. However, look what happened in the case of the older subjects. The older subjects When there was only one distractor, they weren't too bad. And when there's 0 distractors, they were, not too bad in terms of the young people. But when we go to 4 and 8 distractors, the older person's sort time slow down dramatically. And this led to a conclusion that that Patrick Rabbit put in the literature that stood the test of time, and that is that older adults demonstrate an inability to ignore task-irrelevant stimuli. This is seen as a hallmark of normal adult aging, this failure of selective attention that begins to develop, this inability to focus in on task-relevant stimuli while at the same time ignoring, if you will, task-irrelevant stimuli. So this is a very important finding in gerontology, but it begs the question, why is this happening? Why is there this failure of selective attention when we grow older? And as we can see in this next slide, there's really 2 major mechanisms that have been identified, that have been revealed through perhaps hundreds of scientific studies that have been done on cognitive aging. And the 2 major explanations of this deficit in selective attention that accompanies old age that have emerged have been, there's a general cognitive slowing component, and also, more specifically, there's a loss of inhibitory cognitive control. Inhibition in the frontal lobes is one of the major mechanisms of cognitive control in cognitive science and in the neuroscience literature. And it's interesting, if you'll hearken back to our consideration of the aging brain, that one of the dominant themes that emerged was this hypofrontality theme, namely that of all the brain areas, it was the frontal lobes that seemed to be most affected by aging. So it's interesting, isn't it, that on the one hand, neuroscience tells us look to the frontal lobes to explain cognitive aging. And on the other hand, if you will, laboratory science has demonstrated behaviorally that there seems to be a loss of inhibitory cognitive control that is contributing to, if you will, this deficit in selective attention. So what we're going to do now is we're going to look at sort of what types of studies essentially contribute to the idea that there's a loss of inhibitory control. And then we'll also look at a class of studies that reveal the component that general cognitive slowing plays in this, if you will, degradation of selective attention capabilities as we grow older. So first, let's consider the inhibitory control mechanism. And there are 2 experimental sources of evidence we're going to review here. And one is a phenomenon called the Stroop interference effect. We're going to see what happens to Stroop interference as people get older. And then we're going to look at something called negative priming effects. And we're going to see that those virtually disappear in older populations. And as we'll also see, since negative priming effects depend upon inhibition, the failure to observe them is very consistent with the idea of what? That normative aging is accompanied by a loss of cognitive inhibitory control. So let's begin with the Stroop interference effect. Stroop interference is a well-known phenomenon in experimental psychology. And it occurs when the processing of a specific stimulus feature, a specific aspect of a stimulus, impedes the simultaneous processing of another property or aspect of that same stimulus. Now, that's a pretty abstract statement I just made. Let's try to hone in on it a little bit more by looking at a concrete instance of such a situation where one feature of a stimulus can interfere with the processing of another feature of the stimulus. And what we see over here on this slide here are some famous examples of what are called Stroop interference words that appear in the Stroop color word test. And you'll notice that we can read these words quite easily. This word says blue, this word says green, this word says red. And if that was our task, we could perform it pretty quickly. But if we, instead of saying, what does the word say, if our task is to say, what color ink is the word printed in, you can see a conflict is set up in this first word. The word blue is written in what color ink, or in this case, light on your display. It's written in red. So that when you're presented a word like this, a Stroop interference word like this, and your task is not to read the word, but to identify the color of the ink, what happens is you're so overtrained to read words, it happens so automatically and reflexively that you tend to want to say the word blue here instead of the correct response, red, and that there is a strong need to inhibit, cognitively inhibit, suppress that automatic reaction of reading the word. And so what that means is our reaction time typically slows down when we're presented with words and have to name the color of the ink instead of the actual name represented by the word itself. And so the Stroop test really sort of gives us a way of looking at the strength of our ability to control, if you will, to apply inhibition of these competing responses. So it's often used to assess the ability to assert cognitive inhibitory control. And we're going to see what happens when we try doing that in older folks in just a moment, in fact, in this slide here. So what we see here is, if you look at age down on the x-axis, 20-year-olds, 40-year-olds, 60-year-olds, 80-year-olds, and we see what the reaction time is to name the color of the ink for the presentation of the Stroop interference words that we saw in the previous slide, we can see that older people have a difficulty suppressing that, if you will, automatic response to reading the word. And so there's a little bit of an effect in young people compared to the control. I'll explain the control in just a moment. But as we get older and older and older, our ability to inhibit the competing response becomes more and more difficult, as represented by this increased slope in the Stroop word function and this increasing distance between, if you will, the control condition and the Stroop word situation. So what's the control condition? The control condition is you might be shown words where, like this word, blue, green, red, and yellow, these words, where there is no conflict between what the word says semantically and what the color of the ink is. And when you look at reaction time for naming the color in these cases where there's no conflict, you see essentially the slope as a function of age is relatively small. It sort of reflects simple reaction time slowing. Or if you name the color of control objects like these geometric forms, we see that the rate at which the reaction time slows with age is much less for these control situations where there's no need to exert inhibitory control. But in the case of the Stroop interference words, where there is a strong need to exert inhibitory control, we see ever increasing differences between the control and the interference condition as age advances. So we say there's a diminished inhibitory control that accompanies normal aging. That's a relatively straightforward conclusion that we can draw from the results of experiments that use Stroop interference and related types of phenomena. And there's another source of experimental evidence that provides particularly strong evidence for the hypothesis that old age is accompanied by diminished inhibitory control. And the study I want to describe here is one that was done by Hasher, a researcher at Michigan State University, and her students. And in this experiment, she used the negative priming paradigm, which I'll explain in just a moment, to demonstrate the diminished strength of inhibitory control in healthy older adults. And one of the reasons this experiment is particularly interesting is she sets up these conditions in this experiment so that if older people do indeed have weakened inhibitory control, In the second phase of our experiment, the older people should actually do better than the younger people do in this experiment. And that's really kind of clever. Whenever you can actually set up conditions and so that older people have superior performance than younger people, then you really feel pretty confident that you're onto something, because it's so rare that when we compare young and old people on a particular task, the old people do better than the young people. So keep that in mind as we talk about the negative priming paradigm. So negative priming of selective attention occurs when you have a distractor stimulus in the first block of experimental trials in, let's say, a visual search task. And that distractor stimulus during that first phase has to be, if you will, suppressed. It has to be inhibited in order to be able to quickly find the target stimulus. Negative priming occurs when that distractor stimulus in the first phase of the experiment suddenly becomes now the target that you're looking for, instead of suppressing in the second block of the experiment. So what happens in this situation is, let's suppose you do 200 trials on the first block where you're looking for certain letters, a certain target, and the distractor is always some other target, let's say an X, for example. Well, over the course of the 200 trials, what happens is you learn to inhibit that particular stimulus, the X, because it's always a distractor. Now you take a break for 5 minutes and then you do another 200 trials. But this time, instead of looking for whatever that first target was, X now becomes the target. So now you need to find the X among, let's say, some other distractor, maybe K's. What has happened, however, during the first 200 trials is you have developed a, if you will, a bias to inhibit the stimulus that was a distractor on the first 200 trials. So what happens is your ability to find that target that was once a distractor is impaired. So your reaction time slows down, and that's what we mean by negative, if you will, priming. You actually see a negative effect where previous exposure to a stimulus as a distractor causes you to become significantly slower later on in the experiment when the distractor flip-flops its role from distractor to target. So here's a description of a typical type of experiment that would demonstrate negative priming. And let's take a little bit of time to wrap our minds around this. And once we sort of understand what the experiment looks like, what we'll do is we'll examine the results first for young people, and then we'll see what happens when we do the same experiment with older people. So you can see in this case, we start with experiment block number 1. That's what we'll call the priming phase. And in block 1, we have 2 groups. We have one group we're going to call the negative priming group, and we have another group that's the control group. And the stimuli in the visual search paradigm that the control group is going to see is going to be slightly different than what the negative priming group is going to see. So let's first take a look at what's going to happen to the control group. They're going to always be searching for the letter T. And the letter T is always going to be in an array of letters where the distractors are the letter A. So you can see an example of what a given trial might look like here. In this particular trial, 9 letters are presented. 8 of them are distractors, the letter A, and 1 is a T, a target. So in any given trial, there's either going to be 9 letters that are all A's, or there's going to be 9 letters, one of which is going to be a T. And you have to, as fast as you can, when those stimuli are presented, say yes or no, is the target present? So you do several hundred of those trials where A is always a distractor, T is always the target. And in the negative priming group, they're also looking for the same target, the letter T. However, their distractor is different. The distractor in this case is the letter V. So we would see T embedded in an array of V's. And the negative priming group will, just like the control group, maybe do 200 or so trials in phase 1 of the experiment. So what we assume is happening in phase 1 of the experiment is the control group is learning to inhibit A's, because that's a distractor in their case, and negative priming group is learning to inhibit V's, because that's the distractor in their experimental experience. Now we take a little bit of a break, maybe 5 minutes, and then we run experimental block 2. This is the test phase. And in the test phase, all the subjects see exactly the same stimuli. The letter V is the target, and the distractor is the letter X. So we might see something like a V here embedded in an array of X's. But as you see, what's different for the priming group, negative priming group, compared to the control group, for the negative priming group, the V, that's the target now, used to be the distractor. But that's not the case for the control group. So if there's some sort of inhibition that is developed that's going to inhibit the, if you will, the ability to find the letter V, it should only be demonstrated in the negative priming group and not the control group, because only the negative priming group had the previous experience of having to systematically inhibit the distractor letter V. So with that as a setup, let's see what the results are. And what we get is, in this experiment, this is data only from young people. What we see is just what we'd expect. That essentially for the negative priming group, when we look at their search time to be able to find a target embedded into in an array of distractors, we see that they have a pretty efficient search time here. But when we bring them back in block 2, and they now have to search for the letter that was previously the distractor, they have slowed down considerably in the second block of trials compared to the first block of trials, because of that inhibition that developed for the letter V in the first block of trials. For the control group, however, they show the same average search time as the negative priming group in phase 1. Block 1 of the experiment, there's no reason to suspect they wouldn't. But in the second block, since they don't have any, if you will, they didn't have the opportunity to learn to inhibit the letter V, they're just as fast in the second block as they were in the first block. So we have this big difference that emerges in reaction time between the performance of the control group in block 2 and the performance of the experimental or negative priming group. So that difference right there is the way we could measure the magnitude of the negative, if you will, priming results. And that the bigger that difference is, the stronger we would infer the amount of inhibition that was developed during the first block of the experiment. So now we ask ourselves this question down here on the bottom of the screen. What would happen if we did the same experiment with older people? What should the results look like if we assume that inhibitory control mechanisms weaken when we grow older? What we would expect is what? To not see this big difference between the control here and negative priming group. And in the next slide, what I present is the data from the young people in the previous slide who were subjected to the negative priming manipulation. And the red line here represents older people who were subjected to the same negative priming situation. And what you see is a really fascinating outcome. What you see is that in the first training block, where there was yet no opportunity for inhibition to develop and to cause negative outcomes, we see what? That on average, the young people were significantly faster than the older people, and that's not expected. So we see the typical age-related decrement in performance compared to young people. But because the old people were not susceptible to negative priming due to diminished inhibitory mechanisms, look what happens in the essentially second block. The old people don't slow down, but the young people do. So in the second block, we actually get a significant difference between young and old, but this time the old people outperform the young people. Why? Because the inference is they have impaired inhibitory control. And because we've kind of turned the tables and set up a situation where old people are actually outperforming young people, this gives us particularly salient and strong evidence for the role of inhibitory control processes and the weakening of those processes in normative adult aging.